Okay. Hello, everybody. Good morning or good middle of the day, wherever it is that you are. Let me quickly change this. There we go. I hope everybody's having a good end of the week and looking forward to the weekend. We have a long weekend over here in Japan, and I'm going to be spending a little bit of time making some videos for you guys. But before we get into the plans for the rest of the while, just a quick introduction to what we're going to be doing today. Effectively, this will be kind of a biology lesson. I thought that it's useful to know what is happening with regards to the physics that give rise to living things, so that we can understand a little bit more about how the laws of physics and how non-living phenomena give rise to, again, sorry to use the same word, but give rise to the animals and things that we see around us. This also lets us understand what some of the limitations on living things can be. Obviously, it's pertinent to our purpose because we're discussing a lot of living things and discussing what is possible and how things can evolve and change, etc. Before we get into that, though, this is an interesting bird over here. Last week during the live stream, one of the commenters, it was Great Circuit. Gosh, I see his new name now a lot, and then I forget that I after he introduced himself as Great Kobe, I now just associate that with him. Planet Zoidstar mentioned the Hoatzin the Hoatzin bird, sorry, as something that we should look into for something that could potentially be a kind of dinosaur breeding animal. And this Hoatzin bird over here is what you see. It's very, very beautiful and very colorful. It actually reminds me a little bit of the phoenix that we've discussed in the past. So, you've got the red plumage, you've got the the head crest. It's a very interesting bird, very beautiful bird as well. But what makes this bird in particular interesting, also incidentally, I have set this stream up so that I can transfer over to a web browser. What makes this bird particularly interesting is that it is born with a claw. That's that's pretty cool, I must say. We discussed this last week in reference to birds that have atavism and trying to rebreed different types of dinosaurs into the ecosystem. Now, this bird, when it is born, it has this claw structure that you see over there. It's sort of coming off the top of the wing bone there. It's labeled as the distal phalanx and the proximal phalanx, that structure. And it uses this to climb trees. In fact, this is an old 1920s image, just off of Wikipedia, of one of the birds actually climbing a tree using this distal digit. It's pretty much like an Archaeopteryx. Of course, as the bird ages, this bone structure disappears. But at least initially, these baby birds are very dinosaur-like in their construction. Mind you, the relationship that birds have to dinosaurs is sort of like in a side stream. They are very similar to the theropods, but they are nevertheless a distinct category of animal. So that's something to keep in mind. Either way, thank you, Planet Zoidstar, for mentioning that. This was a very interesting bird to learn about. Of course, again, its coloration, for me, being one of the most striking things. Alas, the wings on this one are a bit more dull. Probably indicating that it's female, I guess. But this one over here, my word, look at that. That is a heck of an image. Anyway, now, let me quickly get over to doing the main thing that I want to do today. I'm going to quickly click on a few things here. Give me one moment. Okay, and there we go. So this is, I'm just using Photoshop for this, so please don't feel, you know, too worried when it throws up an error or something that I've clicked something funny. But yeah, one thing I should mention is that I can't quite easily see the chat at this time. The chat is a little bit obscured just because of the way that I've had to set up the screen and whatnot. I will try and keep track of everything with my cell phone over here, but it is still a bit tricky. So just work with me on this note. It's gonna be a fun ride. Now, as I said, what I would like to do is I would like to discuss exactly what is happening with regards to the physical properties that are governing the construction and the formation of living things. That isn't to say that there are, there's only one way to go about this, but you will see some general ideas and limitations form from this. Now, here is where we begin. We start not with talking about living things per se. We start by talking about an atom. Okay. Now, you know that an atom, this classic idea of an atom, is kind of incorrect. But this is basically what it is. You have your proton here in the middle, and you have an electron on the outside. These two things are the building blocks of all the things in existence. And I mean, there's also quarks beneath this. But again, this is basically what we're going to be looking at as the building blocks for living things, because it's at this level where we see electromagnetism come into play. Okay. So, now, as you know, the electron is negatively charged. Let me change my swatch. The electron is negatively charged. And the proton in the middle here is positively charged. And this is what causes them to sort of like each other. They want to move together, and they have a fun time. Of course, as I'm sure you've heard, this is not really a very accurate description of what a atom looks like. In general, it's a tiny, tiny, tiny little dot surrounded by a big probability cloud where the electron floats around. That's a bit more accurate, but this over here is useful for our purposes today. So we're going to be looking at things using this model of the atom, in general. So, with this in mind, we move over to taking a look at the way that this binds to other things. Usually, what you get is atoms forming longer, more complex molecules by the two protons of an atom forming a covalent bond. Now, you've probably heard this in high school physics class, and essentially this is connecting your high school physics class to your high school biology class. What will happen is that these two atoms over here will share their electrons, and they will become bound within a single sort of electron cloud. The bonds can be a little bit different from what I'm drawing over here. This is just for illustrative purposes. But that's not the only way that things can bind. You see, what happens is that we have something called a hydrogen bond. And these bonds usually form with hydrogen, and that's what we're going to be looking at. These bonds usually form on things like water. So you'll have a big oxygen atom over here, and a hydrogen atom over there and there. Now, the hydrogen will give its electron to the oxygen. Hang on, let me take a step back. The oxygen has a greater electronegativity. I think it's 2.5. Electronegativity is the propensity for something to pull electrons towards it. Okay, so that means that when the hydrogen is donating its electron to the oxygen, the electron will spend more of its time around the oxygen. So that generally means that the oxygen will end up being more negative more often, and the hydrogen will be positive more often because its electron is over there, right? And the same is true for this hydrogen. Actually, I have over here, let me see, I have over here a electronegativity chart. I pulled it up just now and then I completely forgot to move it into my work thing here. Okay, let's see. This should help. Think over here. Not that. No, I cannot. Okay, hang on, I got it. So let's take a look at the browser over here, and over there you can see that we have the electronegativity chart starting from a low point on the left hand side and moving up, and then cascading suddenly down. Now, in the top left there, you see there's hydrogen, we're at 2.1. This is how willing it is to pull electrons from other things, and it pulls electrons from all of these things very easily, these ones that are lower than it. And then of course here's oxygen. Oh yeah, sorry, carbon was 2.5. We're going to talk about carbon later, ergo my mistake. Oxygen is 3.5, so oxygen over here is more likely to pull hydrogen's electron over towards it. And that basically creates this imbalance that we're going to be looking at for how living things operate. I'll go and correct the image just now. What's also important is that you'll see that these organic chemicals over here, these things that are non-metals, these tend to have a greater electronegativity than the metals themselves. Even though these metals are very good conductors of electricity, like copper, silver, and gold. Platinum is over here, and there's palladium over there. Iron Man's special suit thing, the PD. Excuse me. Now, everything drops off over here at the noble gases because they have their valence electrons filled. They don't have easy chemical reactions. But yeah, this is something that's important to remember, that generally there's an upward cascade and a pulling towards of electrons as we go further on in the groups here. Okay, so moving back to our discussion over here. Change that to 3.5, my complete and utter embarrassment. So what ends up happening here is that whoopsie, that's a very big eraser. What ends up happening here is that you get a water molecule, and again, we're discussing water mostly because it is relevant to our topic. You get a water molecule that has a slight difference across the entire molecule of, what was it? It was 2.1, I think. For hydrogen, yes, 2.1. That has a slight difference in the charge across the entire molecule. So that means that although the atom itself is ostensibly neutral, a single atom is neutral, the molecule has a different charge across the entire structure. This is like a regular magnet. And this is something that we should try to remember, because a lot of the interactions that we're going to be seeing is this magnetic interaction. And in fact, what I'm going to be describing for the rest of the, I guess, this lecture, that it's going to be, is how this magnetic force between, well, water and other chemicals creates building blocks of life. All fun. Okay. And one more thing to mention is that there can also be within a single atom something called a dipole moment. This is where the electrons, for statistical reasons, all tend to gather at one point, meaning that for a brief moment, this side of the atom is negatively charged and this side of the atom is positively charged. This only lasts a very, very brief moment. Okay? But this leads to things like Brownian motion, where atoms and molecules jiggle around. For reference, this is called a dipole. This molecule over here is called a dipole. Okay? And dipole, P-O-L-E. And basically, it describes what it means. Di means two, and pole means sides. So two sides. It has two parts to the, to its polarity. Okay, so that's essentially what we're talking about here. Atoms have electrons and protons. Electrons are negative, protons are positive. They bind in a covalent bond usually. This is very strong and hard to break, relatively speaking, of course. But when they make this covalent bond, like what we see here in water, they are given a charge. Okay? Usually given a charge. There are cases where their charge across the entire molecule can be even. For example, with hydrogen bonding to itself, H2. And this dipole means that it works like a magnet, which means that it has an imbalance, a natural imbalance, and that natural imbalance lets us see cool things happening. Okay, and here is a nice picture for you while I get everything set up. This is supposed to be my dipole atom with its electrons sitting in weird spaces, not completely spread out across the atom. Let me take this as a moment to quickly check the chat. Okay. Not to worries. No questions. Nobody wants to know anything. That's absolutely fine. You can always ask later on in the actual chat at the bottom of the video. Now, let's talk about dissolution. Dissolution is when we put a chemical into a liquid form of another chemical. So again, let's go back to water, because that's going to be our main thing that we're thinking about here. So you've got your water molecule. Whoopsie daisy. Let me draw on the correct sheet. There we go. You've got your water molecule, and it is connected to yet another water molecule, and yet another one. Whoopsie. Here we go. There's another one. And they're all having fun. And this is water. They don't necessarily have to be this close to each other. They jiggle about. They move between one another. When they are solid, their jiggling stops. That's kind of the second law of the third law of thermodynamics. So, things stop moving. The more you remove the energy from a system, the less things are likely to move. Let me see if I can show off how much I remember. The symbolic logic is... Yes, no, I can't remember the symbolic logic for it right now. But yeah, so basically going back to water, the water over here is binding to each other electrostatically, and they're generally jiggling about. Now, if we take something like sodium chloride, this is table salt, and we put it into the water, what ends up happening is that the sodium chloride is going to dissolve. Now, what does that mean? Well, generally, when something dissolves, it gets spread about inside of the liquid. Okay? So, we usually think that the structures of molecules are very strong, especially with covalent bonds. But in the case of these more simple types of molecules like sodium chloride, the water will actually move in between the two atoms and form an ion. So you'll have sodium ions and chlorine ions. And like this. Let me see here. Sorry, I'm too busy staring at the wall. So you'll have your water over here, and then maybe your sodium is, let's change colors. Maybe your sodium over here is binding there. Actually, let me just quickly check something. Sodium. Okay. So there we go. So the sodium will be over here, and maybe it will bind to, I think sodium is going to bind to the oxygen, if I remember correctly. Okay. And it will bind over there, and maybe you'll have another atom over here. And the chlorine that it is bound to will be binding over here. And most importantly, it is actually physically separate. So your chlorine will be over there. Now, although these two these two atoms are still technically connected to one another, electronically, that is to say, you know, the sodium has lost its electron to the chlorine, they are nevertheless physically separated by a single water molecule here. So this is what it means to dissolve a salt like this. Salt happens when you have a metal, the sodium, bind with a non-metal like the chlorine. Now, there are obviously molecules that do not break down in water. You can have, for example, a protein. When this is introduced into water, all that happens is that the water moves around it and separates it physically from other proteins. Sugar also does this because it's a very, very long, wiggly chain, and the water will just surround it rather than breaking it in half, as we see over here, making the ions. In this case, the chlorine would be negative and the sodium would be more positive. If you take a look over here at that chart again, you'll see that sodium there on the far left, just under hydrogen. Let me quickly go to the browser myself. That sodium over here is 0.9 electronegativity, which means that it has this much pull of the electron, versus chlorine over here on the far right as 3.0. So generally speaking, the chlorine is really going to pull on the electron from sodium. So when they're physically forced apart by the water molecule, he's going to keep the electron and he's going to be positive. Okay? Very simple, very easy to understand. And I want to emphasize that, you know, it's not cheating to look up a chart, because a lot of people feel that you should know the stuff off by heart. It's okay if you have to look it up. The main thing is understanding how to use the information, not necessarily what the information is, especially in these days of AI and whatnot. So we've got an example over here of sugar being separated by, well, not separated by water, but surrounded by water, and it's separate from its other sugar friends by water molecules. Whereas in the case of sodium chloride, the sodium chloride would be actually have individual atoms made into these ions over here, where they're missing electrons essentially. Okay. Now, this solution is important because understanding that water can do this and understanding how water's own electropolarity causes things to separate is an important part of how we start to build living things. So, for example, if we look again at the water molecule, there it is, and we remember that the hydrogen here is, because of the electronegativity of the atoms, the hydrogen will be positive and the water will be negative, the oxygen will be negative. Necessarily, the sodium is going to want to go to the hydrogen. It's not going to want to go over here. Yes, hang on. Am I thinking of this right? I feel like I'm doing this wrong. It's not necessarily going to, either way, they're going to separate, and one atom is going to go to one side of the polar bond, whereas the other atom is going to go to the other side. No, I'm wrong. Yes, okay, no, I'm wrong. I just remembered. No, the oxygen is going to bind to the sodium, and the chlorine is going to be binding to the hydrogen. I completely forgot my electronegativity. My mistake, everyone. My embarrassing mistake. But that's okay. So here we go. The chlorine is going to bind more to the... the sodium is going to bind to the oxygen. I had it right just now, dang. The sodium is going to bind to the oxygen, and the chlorine is going to bind to the hydrogen. Okay, there we go. Now, that's all good and well, but this means that you can actually get things to separate, and separation is what makes living things. Okay, so here's oh yeah, and what's important to remember is that not all molecules, when dissolving in water, actually separate like this. So there you go. There's some sodium molecules and whatnot getting ready to do stuff in water. Now, there is an important phenomena to understand, and this is the concept of hydrophilia and hydrophobia. Essentially, hydrophilia means something that loves water. Philia is the love of, and phobia is the fear of. These words don't necessarily mean that these atoms have feelings or emotions, but they do let you understand how these will behave in a system. So we have chemicals that are hydrophilic, like sodium, that want to absorb water or want to be dissolved in water, and chemicals that are hydrophobic, like oil, that would prefer not to go near water, will not dissolve in water. And this is because of that electrostatic charge that the molecules have. Generally speaking, if something has a neutral charge, is non-polar, then it cannot be dissolved by something that is polar, and vice versa. So if you were to, for example, put water into a mixture of oils, you would find that it floats on top. The rule is that like dissolves like. Okay? So generally you can dissolve oily non-polar mixtures into oily non-polar mixtures. This is why we can, for example, put perfumes together, because generally a lot of the things are non-polar. They're mostly these long carbon chains. Okay, carbon chains generally are polar, but you do get some that are mostly non-polar. Very few though. So now to intermolecular static forces. Now, we have a dipole interaction effect. What we're discussing here is the basically dipole interactions. And here we go. Carbon binding into water. Now, let's see here. What you have with living chemistry is a bunch of carbon atoms. Okay, I'm sure you've all heard, whoopsie, wrong color. I'm sure you've all heard that carbon is the backbone, the building block of life. So we've got carbon chains and they're all happy. Typically, the carbon chains that we draw are assumedly connected to hydrogen, hydrogen, and hydrogen, but this takes a long time to write, so we just assume that the carbon is all saturated with hydrogen. Okay, this is actually called a saturated fat when we're talking about fats and lipids, etc. Because if all of the hydrogen bonds are full here, then there's no more room for any other bonds to be made, ergo saturated, it is full. Unsaturated fats would have a double bond at a carbon, one of the carbon points here, and then the tail would be like that. Okay, this is propene that I just drew over here. So there we go. And then we can put maybe an hydroxy group over there and then make propenol, just to be funny. No, I'll remove that because that will confuse everyone. So these carbon chains over here can have, like with water, a point where they are more electronegative than at another point. And of course the water will, move around them. You'll get some water floating here and floating here and floating here, all over. What's important is that with the carbon chains, there can be points along a very long and complicated chain where it is, say, negative over here, but positive over here and here, but then again negative over here once more. This means that you will end up with a lot of different kind of bonds happening with the water over here, and the water will, in this case, make it jiggle in a particularly strange way. And this also means that with these long carbon chains, again, turning eventually into proteins and amino acids, etc., that they can bind in different ways to each other. Okay, the same protein, the positive sides over here will not want to connect to the positive sides on the opposite side. Instead, they will preferably go to the negative sides here, and that makes a particular shape. This is eventually going to turn into protein folding, but we're not going to get into that today. We'll leave it at simple things for now. So, moving on. What happens then is that we end up with a long carbon chain that has water all over it, and it's jiggling around and it's having a good time. It can also bind in various ways to other carbon chains, and it can make very complex carbon chains. And some of the complex carbon chains that we would see it making are things like monomers. Monomers are the basic building blocks of polymers. These all eventually build up when they bind in these ways that I've described over here to become the more bigger versions, the things that actually make up life. For carbohydrates, the monomers are monosaccharides or sugars, like glucose, for example. Glucose is basically in everything. It's one of the most efficient ways that plants store and move energy around. Sucrose is a polysaccharide. That's your sugar in your kitchen, and it's made out of glucose and fructose. And you get all sorts of fun things for storing energy, for building things, etc., etc., etc. So, when you are eating food, actually what your body is doing is breaking these things down, breaking a long carbohydrate that you don't really, your body doesn't necessarily know what it is or whether it's useful to you, but it breaks it down and turns it into these monomers again, or maybe it turns it into slightly longer monomers, or like not yet a polymer, but what's the word? Bimer or something. I forget what the word is, but it basically means like two of these monomers together. This is why, if, for example, you eat something, a living animal or a plant, even though you're consuming its DNA, the DNA doesn't actually affect your own living body. It doesn't actually interact with your cells at all. So that's something to remember. Now, going on from there, we have these chemicals that are interacting with the water and interacting with each other due to these electrostatic forces. What ends up happening is that the carbon will bind with the water and it will jiggle about. And this is very important for moving things closer together, because if everything was just static, you wouldn't end up with how can I say, you wouldn't end up with just life happening at all. And what's important is that because of this difference in charge across a monomer or a polymer, you can end up with one part of the chemical preferring to be near water, and one part of the chemical, say the bottom, preferring to be away from water. Well, this isn't actually true. It doesn't want to be away from water per se. This side is just pulled towards the water molecules, and as a consequence, this side over here is pushed away. Think of it like the magnets at the beginning. It's not that the here we go. It's not that the magnet is actually pushing the side away necessarily. It's that that's a bad example. But basically, one side is getting pulled towards the water, wants to be near the water, and the other side wants to be with the not with the water, and so this causes things to align. Now, that's basically how carbon interacts with water, and well, carbon chains can interact with water, and that also then gives us parts of carbon that separates and moves about. Check the chat. Okay. Again, if you guys have any questions, do let me know. I know it's a bit complicated and maybe not easy to follow, so that's why I ask questions. Now, hydrocarbon. Just generally, very, very briefly, hydrocarbon chains cause things like carbohydrates to form. And this is useful to know because basically this is now getting to the structures. You'll have carbon, oopsie daisy, wrong layer. You'll have carbon connecting all along here. And these will maybe connect to other carbon chains over here. Okay. And this will form the structures that you need to survive. These have a tendency to wind over each other, especially if the charge across them is even. If the charge across them is not even, then you end up with those weird little fishtail kind of things that are happening. Importantly, is that other atoms can connect to this. So you may have, for example, over here, an oxygen. The oxygen can only bind to two things. I won't get into that right now, but it generally only binds to two other atoms because it has two valence electrons. Again, that model is not 100% accurate, but it is fit for our purposes. And actually then the little bend will happen over here, but that's beside the point. Then you can also put inside of here additional, when the charge over here is a bit weird, you can put in other atoms that might fit into this corner over here without actually being covalently bonded, as these are. The bond between this atom and the entire molecule here can generally be strong enough to hold that molecule. And this is very important again because it lets us do things like have blood. Blood is 4 proteins over here with a little iron atom, and the iron attracts oxygen inside the blood cell. So they can be within the blood cell, but without actually properly binding to the blood in a way that is difficult to remove. I should at this point mention that this covalent bond over here is actually very difficult to break. It requires a lot of energy. However, a bond like this, these hydrogen bonds, these electrostatic bonds, are relatively easy to break. And as a consequence, this is also why life is as easy to mutate as it is. I'll get to a little bit of a discussion about that towards the end, but I just want you to remember that this bond here is weaker than this bond. Okay. And there's 2 hydrocarbons kissing one another through water. Yay. Let me quickly check chat. Okay. So, we now have a general understanding of how the hydrocarbons interact with water and how they interact with each other. And this is now the important part. This is also the last thing that I'm going to discuss, which is how the lipid bilayer is formed. The hydrocarbons are floating around, and they're having a fun time. Mostly because they're getting jostled about by electrostatic forces and all sorts of stuff. This means that they're going to be able to bump into each other. When they bump into each other, they will bind and they will form big structures. Now, you'll have a bit of glycerol over here. Whoopsie, wrong layer again. You'll have a bit of glycerol floating around, and it will meet 2 lipids, and it will make a little tail. And he doesn't just want to meet 2 lipids and bind to them. He wants to meet a phosphor chain. Any kind of phosphor chain will do, something that has phosphorus in it. Okay. These are 2 fatty molecules. So basically, these are the ones that have their tails straight. They're not the bent ones. Okay. These are saturated most of the time. In fact, all of the time, if I recall. So, one of over here there'll be a sort of a phosphate group, like a little head, and it will bind to the glycerol over here. Now, this side over here has a charge, and this side is nonpolar. So this nonpolar section, if you remember, is going to be hydrophobic. It is not going to want to go near water. Whereas this part over here wants to go near water. So you'll end up with this little structure with its 2 feet, and it will point towards some water molecules sitting over there. Okay, and if there's no water, then these little legs will point up away from the water. Let's say there's air over here, for example. Because of the van der Waals forces, that's the actual electrostatic jiggling that I described before. These things actually will meet up, and they will make sort of a chain. Okay, then they generally bind to each other like this in a chain. But of course, if they're in water, which they usually are, the little feet are not very happy. They don't want to be near water. So what usually happens is that another set will come over here. Okay. And now the feet are happy because they're pointing towards each other where there is no water over here. And these heads are happy because they're pointing towards the water. And all along here on this inside area, there will be no water. There will be no water molecules at all. Maybe one or two, just due to random chance, but generally there will be none. And this is the cell membrane. This is how your cell membrane is formed. Going from electrostatic forces moving things around to this larger, I would say it's a coherent structure, but larger non-intuitive structure over here creates this wall. What ultimately ends up happening is that these little things will bind to each other in a circle like this. And in actual fact, this circle is pretty solid. And now you have effectively an oil drop where there's water on the inside over here, H2O, and water on the outside, H2O. The heads over here on the inside section are happy because they're pointed towards water, and the heads over here are happy because they're pointed towards water on the outside, and they make this ring. And this is not just a circular ring, it is in fact a 3D sphere. And it's got a little shadow over there. I don't know why I did the shadow on the back over there. Let's put that over there. Okay. And all of these things will have a good party and they'll have a lot of fun. Let's quickly erase my shadow. Now, what this means is that we have a space where we can do chemistry that is separate from the environment around it. And that means that we can control the concentrations of chemicals without them dissolving into the surrounding environment. If you remember, the sodium chloride had a tendency to get separated and dissolve, and they all get pushed around. What's very important to understand about this is that the chemicals in a volume, in any amount of water, will get pushed around and separate and go to fill the vacuum, as it were. Whereas if we have this structure here, we can have a higher concentration inside the structure than we do on the outside. And if we have a higher concentration of something, then we can start to do interesting chemistry with it. Then we don't necessarily need to do chemistry to the entirety of the water around it. We can do just a bit of chemistry in here. And starting from the structure here, starting from it forming from electrostatic forces, we then have the ability to make life. Now, I'm going to end this very disjointed discussion over here, but basically understanding that we go from single atoms. Let me quickly go down over here. Whoopsie daisy. We go from single atoms over here with their van der Waals charges, where sometimes the electrons just happen to meet up over here and leave the side positive and the side negative and causing jiggling about, as well as binding to one another and leaving an entire molecule with polarization, all the way up to something like this, where the water is causing certain parts of this entire structure to move away from it and certain parts to move towards it, or rather not to move away, but just certain parts to move towards. We end up with a structure that lets us have a higher concentration and a lower concentration. And again, remembering how dissolution works, that we can separate things, the sodium, into water as well as sugars, etc. And then that's life. And yeah, there's your lipid bilayer sucking up water. It does have structures to help it. It does have things that let it work on how to manage the concentration of water on one side and water on the other side, and salt on one side and salt on the other side. But that's basically the simple and straightforward idea. So yeah, voila. with understanding that, we're able to more completely know how living systems work and what is possible. Specifically, we understand why water is necessary for life and why it is a little bit difficult for us to justify that life could exist without water. So, for example, if we go back to the electronegativity chart over here, you will see that generally things over here are more likely to you need these inorganic chemicals so that you can actually have this electrostatic valence, a difference between two parts of a molecule. Okay. And you'll also notice that something like ammonia, which is made with nitrogen, does have the potential to function in the same way as water. See, it's got an electronegativity of 3.0, whereas oxygen has 3.5. Although it does have the potential to act like water, it won't necessarily be as strong, just because the difference, the electrostatic difference, will be just a little bit smaller. As you can see, 0.5 difference. Okay, so that's why, again, although it is possible to have alien life with an ammonia environment, generally you need to have that electrostatic valence. This also lets you know that you do need some kind of liquid. Hypothetically, a gas and a liquid are, for all intents and purposes, very similar, but a liquid generally is a gas, just more compressed down towards, and close towards the distance between the atoms and the molecules is less. So you need that to sort of force the other molecules and the other proteins and the other structures together and create that Brownian motion that we were talking about in the van der Waals forces. So it generally has to be a liquid. And this is the reason why we think that we should have liquid water on other planets for there to be life. You can't just, imagine, oh, what if we had a chlorine ocean on a planet, and would life form from that? Generally speaking, from the physics that I explained just now, again, I hope that was simple to understand at least, you'll see that there's not as much pushing and pulling and jiggling and tugging that occurs. There's no incentive for this incentive is the wrong word. There's no electrostatic forces causing things to form coherent structures, especially again with those chemicals that have a charge that is even across them. This is why for the noble gases, the helium, neon, argon, krypton, xenon, and radon, they tend to, even though hypothetically at low enough temperatures they could form a liquid, they won't necessarily create any life in them because their charge is just flat. Okay, so that was a very long and awkward discussion, but I hope that you had a fun time learning about that very, very basic step from how we go from atoms to the start of living things, and especially the start of living structures. If you guys have any questions, please feel free to send them, and you can post them in the chat also afterwards. I will go back a little bit just because there were a few people that would appreciate it. Just one more time, looking at the bird. This bird was mentioned last week by Great Sir Kirby. It is called the Hoatzin bird, and it is born with a claw. And this is in reference to us talking about dinosaurs and atavism. So yes, that's pretty much that, as just a final sign-off and a reminder. I hope that makes sense, and I hope you guys can understand what it is that I consider when I'm thinking about what might be physically possible and naturally possible when doing reifications and stuff like that. So with that, we've been at it for a while, and maybe I'll try and revisit the topic a little bit later. Next time, I plan to talk about this a little bit more detail. These living structures, and again, how we go from the fundamental forces into living structures. And yeah, I need to plan that discussion a bit though, so we'll wait for it for another while. Otherwise, thank you for joining us, joining me, and have a good weekend, guys.